Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled On-Site Pharmacy Compounding for Your Phase 1 Clinical Study. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, webinars are designed to be interactive and work best when you're involved, so please feel free to submit your questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen, and if you require any assistance along the way, please contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Celerion, who developed the content for this presentation. Celerion, a global leader in early clinical research services, offers a unique combination of medical expertise, clinical operations experience, and scientific excellence that gives their clients the confidence to make fast, accurate decisions about their drug development path. For 50 years, Celerion has leveraged the latest operational concepts and technologies to execute safety and tolerability, pharmacokinetic and pharmacodynamic studies in highly controlled clinical environments, offering feasibility, data management, biostatistics, clinical monitoring, and bioanalytical services. Now, I would like to introduce our speaker for today's event. Dr. Foster is the Director of Global Pharmacy Operations at Celerion. She is responsible for overseeing all 3 Celerion pharmacies, as well as ADME studies from a pharmacy, licensing, and total radioactivity reporting perspective. She has worked on over 100 ADME studies with all varieties of formulations and challenges. Dr. Foster graduated from the University of Nebraska Medical Center in 2001 with a PharmD. In 2016, she completed the Authorized Nuclear Pharmacist program from Purdue University. Now, before we begin, we have some poll questions for the audience, so you can interact with these questions that you should be seeing on screen right now by selecting on any of the answer that you see in front of you and then clicking Submit. The question that we're asking you is, what environmental controls are required when working with most investigational API? Your answer options are negative pressure, segregated area, PPE, or all of the above. We'll give everyone some time to consider their answer to this question again is, what environmental controls are required when working with most investigational API? It looks like most of you have gotten your vote in, so thank you very much for participating. Let's look at the results that you have shared with us here today. 72% of you have selected all of the above, 11% each for PPE and segregated area, and then 6% have selected negative pressure. Thank you very much for participating. We've got one more question before we head into our main body of today's presentation. This question would like to know, true or false, investigational products administered by subcutaneous injection or IV infusion are required to sterilized before administration. We'll give everyone some time to read this question. Consider your answer as it best applies, as you think it best applies. The question is a true or false question wondering investigational products administered by subcutaneous injection or IV infusion are required to be sterilized before administration. It looks like most of you got your vote in. Let's take a look at your results, and 98% of you, 96, have said true, and then 4% have said false. So, thank you very much for your time participating in this. We're going to be hearing a lot of great information along this line for today's presentation. So let's go ahead and hand things over to our speaker, Dr. Jennifer Foster. You may begin when you're ready.
Thank you for joining me today on this discussion in extemporaneous compounding in phase 1 clinical research. I will cover several areas today, including challenges and trends in early clinical research. There are a lot, and they are changing all of the time. I will also give an update on USP, the most recent guidelines, and how Celerion adheres to these guidelines, including our pharmacy facilities and training programs. I will discuss the advantages of extemporaneous compounding, when it is appropriate, and when GMP should be used. Finally, I will touch briefly on different study designs Celerion can help with compounding, including ADME studies. Let's begin with some common challenges and trends in phase 1 clinical research. Over the past 20 years, I have heard the same challenges from our clients over and over. Number one, the drug supply is limited. Can you deal with small quantities of drug or API? The short answer is yes, absolutely. There are many creative ways to deal with small amounts of drug supply, including creating smaller batch sizes for oral and IV formulations or sharing vials for IV formulations. The next challenge is formulation development and manufacturing timelines. True manufacturing can take months, whereas compounding is done in real time, within hours to days of administration, saving significant time. Finally, fast data is always a must. Compounding allows for data to be turned around quickly. Trends in phase 1 clinical research also support the use of on-site compounding. Compounding allows doses to be adjusted easily without further manufacturing needs. If a dose needs to be adjusted, this can be done easily and taken care of at the pharmacy level. Highly potent compounds are more and more common, so having the appropriate engineering controls in place to deal with API compounded formulations is essential. Overall, the push for shortened timelines is ongoing. This is industry-wide, and compounding helps to shorten overall timelines. There are many efficiencies of on-site compounding. First, dosage forms can be easily changed and adapted for human administration. This can include an API and capsule, as well as oral solutions and suspensions. Small doses can be administered, which is especially important in a first-in-human trial design. Essentially, compounding allows for dose customization. Of note, all excipients sourced by Celerion are of the highest grade. These grades can include a GMP grade, a USP grade, or an NF grade. Excipients are received based on their certificate of analysis and accounted for on Celerion drug accountability forms. Now I would like to dive a little deeper into the advantages of on-site extemporaneous compounding. First is timing. GMP manufacturing takes time, typically at least 3 months, and in today's world, that could be much longer. Delays are common and sometimes unavoidable. On-site compounding is a much faster and tailored to the project at hand. Celerion pharmacists work with their clients to ensure fast and, most importantly, accurate compounding. The next advantage that I would like to touch on is cost. GMP manufacturing is expensive, especially if you are dealing with sterile products. Celerion is able to handle both sterile and non-sterile compounding at a fraction of the cost that the client would pay for GMP manufacturing. As I mentioned earlier, drug supply is often an issue with phase 1 clinical research. Therefore, being able to adjust one's batch size to account for the limited amount of investigational product is a large advantage. We are able to receive small shipments and/or multiple shipments and store the product appropriately. GMP requires large-scale batches and retains, whereas on-site compounding does not. The last differentiator is stability testing. GMP requires ongoing stability testing. On-site compounding does not have this requirement because the preparations are done in real time and administered shortly after preparation. I am now going to move on to an overview of the USP, the United States Pharmacopoeia. USP is an independent, scientific, nonprofit organization focused on building trust in the supply of safe, quality medicines. Celerion follows the most updated version of USP, which was released in 2019. All of our SOPs and training materials adhere to this version. USP 795 covers non-sterile compounding guidelines. This would include oral formulations, topical formulations, overall anything that would be considered non-sterile. USP 797 covers sterile preparations, including IVs, subcutaneous preparations, and inhaled preparations. USP 800 covers all extemporaneous compounding, including hazardous compounds, with the overall intent of the chapter to protect the worker. Most Phase 1 investigational products fall into this category simply because testing has not yet been done, and therefore products are handled as hazardous. In 2019, the USP released revised chapters of USP 795, 797, and the initial chapter of USP 800. The major revisions of USP 795 include the following. There needs to be a dedicated compounding space with detailed cleaning requirements. Any powder manipulation should be taking place within a containment device. All products should be received with a safety data sheet as well as a certificate of analysis. The requirements of the master formulation record, or what could be considered the recipe, are outlined in this chapter. There is a clear formula for determining beyond use dates. Training is rigorous and required to be overseen by the designated person. Lastly, a quality management system is required. The changes to the 2019 version of USP 719 were also extensive and changed a lot of the previous terminology. What was formerly known as low, medium, and high-risk sterile compounding is now known as Category 1 and Category 2 sterile compounding. There are more stringent controls on environmental monitoring, including monthly surface samples throughout the sterile environment. Similar to USP 795, training is more rigorous and competency must be completed every 12 months, including a written test. The requirement of a designated person is also required, as well as a quality management system, which is very similar to how it is written in USP 795. The layout here shows the different environmental requirements for the specified compounding areas. The USP 795 area is not required to be classified. The USP 797 area is required to be a positive pressure area with greater than or equal to 30 air changes per hour, approximately 20 degrees Celsius, less than 60% relative humidity, and it runs on recirculated air. The USP 800 non-sterile area is required to be negative pressure, greater than or equal to 12 air changes per hour, and runs on fully exhausted air. The USP 800 sterile area is required also to be a negative pressure area, have greater than or equal to 30 air changes per hour, approximately 20 degrees Celsius, less than 60% relative humidity, and like the other 800 area, runs on fully exhausted air. What that means is that those areas are vented to the outside. As I mentioned previously, the 2019 version of USP 797 updated the classification of sterile compounding from low, medium, and high risk to Category 1 and Category 2. Category 1 compounded sterile products include products whose beyond use date is less than or equal to 12 hours at controlled room temperature or less than or equal to 24 hours refrigerated. These products may be prepared in a segregated compounding area and do not require sterility testing. Category 2 compounded sterile products have a beyond use date of greater than 12 hours at controlled room temperature or greater than 24 hours refrigerated. They must be prepared in an ISO 5 primary engineering control within an ISO 7 buffer room within an ISO 8 anteroom. These products also do not require sterility testing if the appropriate beyond use date is assigned. Training to be compliant with USP 2019 is extensive. Personal qualifications include hand and hygiene garbing, gloves fingertip sampling, media fill testing, and requalification, including a written test. Media fill testing at Celerion is done for both sterile to sterile compounding as well as non-sterile to sterile compounding. All of these activities are performed for all pharmacy staff at Celerion every 6 months. Celerion's compounding suites are certified by an outside vendor every 6 months. The suites are certified to the requirements of USP 2019. This includes having the suites and primary engineering controls certified. Certification includes testing of all the HEPA filters, viable air sampling, as well as viable surface sampling. Additionally, monthly surface sampling is done in-house. Cleaning of our compounding suites is one of the most important activities that must be done correctly in order for us to continue to pass our semi-annual and monthly certifications. Cleaning requirements for the different areas of the compounding suites are shown here. Primary engineering controls are cleaned most frequently as that is where the most critical work takes place. The ISO 5 primary engineering controls are cleaned at the beginning of each shift, between products, every 30 minutes, after any spill, and when contamination is expected. Counters and work surfaces are cleaned daily, with trash being removed at the end of each shift. It is imperative to remove all standing water from a sterile environment. The rest of the areas are cleaned at least monthly. All cleaning must be thoroughly documented appropriately. The master formulation record is a detailed record or recipe used for detailed documentation of the steps of how a formulation is to be compounded. According to USP, the master formulation record must include at least the following. It must have the name and the strength and activity of the dosage form. It must have identities and amounts of all of the components. The container closure system needs to be included. Complete instructions of how the preparation is to be included, including the equipment, physical descriptions of the product, the assigned beyond use date, labeling requirements, quality control procedures, and information for reproducibility. USP 800 was new in 2019 and caused a lot of facilities to revamp and remodel their compounding areas, including Celerion. USP 800 was designed to protect the worker while handling hazardous drugs. It requires a designated area for receiving and unpacking, which must be neutral or negative pressure. Storage areas, non-sterile and sterile compounding areas must be negative pressure. There must be a hazardous communication plan, and compounding hoods must be placed appropriately. All equipment and devices need to be decontaminated and cleaned. If the equipment is being used for sterile compounding, the equipment must be subsequently disinfected. Next, I would like to discuss GMP manufacturing versus extemporaneous compounding. GMP is defined as a system ensuring products are consistently produced and controlled according to a set quality standard. Mass production or processing of drug products for the purpose of resale. Extemporaneous compounding is defined as the preparation, mixing, altering, packaging, and labeling of a drug in accordance with a licensed practitioner's medication order for an individual subject. So the question is, which one do you need? The FDA issued a guidance in 2008. The guidance was issued because there are frequent questions around the topic of GMP in phase 1. The guidance states that drugs used in phase 1 are exempt from complying with GMP. The guidance applies to phase 1 investigational drugs, whether they are manufactured in small or large-scale environment, because phase 1 clinical trials are typically designed to assess tolerability or feasibility for further development of specific drug. The intent of the guidance is to have drugs produced in scientifically sound environments without all of the strict environments of GMP. The FDA has issued regulations governing IND products to protect human subjects enrolled in clinical trials. This is done through the CMC section of the IND. The FDA reviews the submitted IND to determine whether the phase 1 investigational drug to be used in the clinical trial is sufficiently safe to permit the trial to proceed. Here I have outlined some more basic differences between GMP and Phase 1 environment to show how the guidance is used in our environment at Celerion. A Phase 1 pharmacy is a highly controlled environment with a relatively small number of employees. Investigational product is prepared for specific protocol for specific subjects. Batch sizes are usually very small and beyond use dates are short. In contrast, GMP is required for large manufacturing needs. Batch sizes are large and distributed in bulk supplies. Facilities are large with multiple staff members. Expiration dates are assigned for the products to be used well into the future. When it's time to place your compounding study with a trusted contract research organization, there are several factors to consider. You will want to make sure the facility is well designed with proper engineering controls in place. The certifications should be verified to ensure they are appropriate for the type of compounding that is going to take place. Robust and thorough SOPs are a must for all compounding procedures. There should be an established quality management system, and staff should be highly trained and knowledgeable. Celerion has dedicated pharmacies and pharmacy staff. The pharmacy areas are secure with dual limited access to only pharmacy staff. The pharmacies have continuous environmental monitoring as well as a DEA license. Celerion operates under global SOPs. All activities in the pharmacy are verified by 2 pharmacy personnel. Celerion has proprietary data capture system called ClinQuick, which is integrated with the pharmacy systems. We also have a global quality management system. Pharmacy storage areas have controlled temperatures that are in line with USP. The temperature ranges are listed here and include room temperature, refrigerated conditions, -20 C and -80 C. We have continuous temperature monitoring with PDF reports available at any time. Rotating pharmacy staff are always on call to handle any excursions. Refrigerators and freezers are on backup generator. The compounding suites at Celerion are monitored by the OSHA View system. This system is able to monitor temperature and humidity and pressure. PDF reports are also able to be generated for this system, similar to the previous ViewLink system. Phase 1 protocols have a variety of formulations of investigational product. The ability to handle different varieties is essential to completing a successful study. Traditional formulations would be instances when bulk tablets or capsules or solutions would be sent to the CRO and then dispensed as directed by the protocol. Non-sterile and sterile extemporaneous compounding typically involves the use of API and excipients to develop a final formulation with the use of a master formulation record. ADME, or radio-labeled studies, are almost always extemporaneously compounded and can be sterile or non-sterile. In order to run an ADME study, the CRO must have a radioactive materials license. Over the last 2 years, Celerion has invested in the Lincoln and Phoenix sites to build USP-compliant compounding suites. Both sites have a dedicated space for compounding non-hazardous and hazardous drug for both sterile and non-sterile formulations. Furthermore, the Lincoln site is able to compound radioactive formulations for ADME studies. Both sites have a variety of equipment, including biological safety cabinets, balances, mixers, sonicators, easy pet pipettes, stir plates, hot plates, pH meters, any kind of compounding equipment you can think of. Both sites follow global SOPs standardized to the 2019 version of USP. Here are the layouts of the compounding suites in both Lincoln, Nebraska and Phoenix, Arizona. The Lincoln facility diagram is on the left, the main difference being the ADME suite in blue. The rest of the layout is similar across sites and includes hazardous receiving storage, a drug formulation room for non-sterile formulations, and a sterile suite including an anteroom, a non-hazardous sterile compounding suite, as well as a hazardous sterile compounding suite. An overview of the Celerion engineering controls are listed here. The engineering controls are in line with current USP. Each specific compounding suite is listed along with the pressure requirement, positive or negative, the ISO requirement, the primary engineering control within the room, whether the room is sterile or non-sterile, and the air changes per hour required. Both Celerion US compounding suites have been certified, most recently in February of 2022. The full reports are available for review during audits and site visits. The overall certification certificates are shown here. Now that we have talked about the facilities and equipment that Celerion can offer, I would like to discuss specific pharmacy services, including staff training and types of studies we can help to conduct. Celerion has dedicated pharmacy staff consisting of PharmDs and registered technicians with over 40 years of phase 1 experience. We have an authorized nuclear pharmacist to run all ADME studies and assign a lead pharmacist to each new study awarded to Celerion. The pharmacist will be involved with the study from startup to closeout and available for any consultation needed. All pharmacy staff are cross-trained on all protocols and all pharmacy functions. Celerion provides comprehensive training program for all pharmacy staff. Internal training consists of robust SOPs as well as hands-on sterile training as well as non-sterile training. External compounding training consists of annual online certification through Critical Point, which includes training on USP 795, 797, and 800. Hazardous drug training consists of a respiratory protection program with annual external training and the use of PAPRs, or powered air purifying respirators. Study-specific training includes protocol and pharmacy manual reading, as well as mock preparations as required per protocol. Internal training consists of a host of required reading materials, including SOPs and the actual USP chapters. Media fill testing and glove finger testing is required. All of these activities are documented and overseen by the designated person. Once the employee has successfully completed these activities, they are ready to compound. Recertification is required every 6 months. The need for several different types of formulations has arisen over the years. Celerion has experience with a lot of different types of formulations, including oral solid dosage forms, API in capsules, sterile to non-sterile, complex oral solutions, emulsions, enemas, suppositories, liquid-filled capsules, over-encapsulation, powder and bottle, taste masking, as well as formulation blinding. We are also always willing to explore new opportunities and enjoy learning with and helping our clients. The typical compounding process is shown here. The master formulation record is created in conjunction with the sponsor, and then, if required, a mock prep is completed. If any changes are needed to the master formulation record after the mock, they are made and approved by the sponsor, and the updated master formulation record is used for the actual clinical compounding, typically done on the day of or the day before dosing. From there, the study execution commences. First-in-human trial designs are a very common time for compounding to be used. We will often fill capsules, make solutions or suspensions, all because it is very easy to adjust the dose as the dose escalates quickly in these types of trials. This will also allow for the multiple ascending dose to run concurrently without delays for manufacturing, and even the food effect can take place during this time. Celerion can receive non-sterile API and formulate to a sterile formulation. This is considered Category 2 compounding. Typically, the sterilization is done using a 0.2 micron filter, which is filter integrity tested using a bubble point tester. Sterility testing is not required if the beyond use date is less than 24 hours. Endotoxin testing is done on site and the results can be available prior to dosing. An example of what the readout from the endotoxin test is shown here, and this will be kept within the pharmacy records. Blinding is something that we often help with. We commonly deal with the question of how blinding is done and have become very creative with maintaining the integrity of the blind. All blinded pharmacy documents are kept within the limited access pharmacy. Once the product leaves the pharmacy, the label that is on the product is a blinded label. To maintain the blind, we have opaque bottles that can be used for tablet dosing. Syringes can be blinded with tape, and IV bags and tubing can be covered to ensure everyone except pharmacy staff remain blinded. Taste masking is also common. This can be done for a couple of reasons, either to prevent unintentional unblinding or to improve palatability. Listerine pocket packs can be administered pre and post dose, and these will basically blast out any taste that the formulation may give off. Another option is to add a product called Bitrex to the actual formulation, which will take the bitterness away. Next, I'd like to talk about the ADME trial design. ADME studies are a type of study that Celerion specializes in and has a great capacity to execute. Celerion has held a radioactive materials license with the state of Nebraska for over 40 years. Typical ADME study designs fall into one of the following categories. The traditional ADME, absorption, distribution, metabolism, and excretion study, are defined by the dose of radioactivity, typically somewhere between 50 and 450 microcuries, determined from the animal dosimetry data. The dose can be administered oral, IV, or sub-Q. The objective of this study is to know how much radioactivity was administered and to collect the radioactivity in blood, urine, and fecal samples. This will determine how the drug is eliminated, either renally or hepatically. Microtracer studies are another study design where a small amount of radioactivity, usually around 1/100th of the amount of a traditional ADME, is administered along with a cold dose of the drug and then followed through the body to determine absolute bioavailability. Finally, there are study designs where both of the above are combined into one protocol, either with one group of subjects in 2 dosing periods or 2 groups of separate subjects. Typical extemporaneous compounding for ADME studies includes filling capsules, either API or liquid filled, solutions, suspensions, or IV or sub-Q. All radioactive formulation concentrations are verified by liquid scintillation counters and doses are weighed prior to administration. Residual in the dosing container is accounted for, so the dose of the radioactivity administered to the subject is very accurate. I would also like to mention that we have recently expanded our ADME lab in Lincoln, Nebraska. We have the capacity to run multiple ADME studies with a precise execution, having the clinic, pharmacy, and ADME lab all in the same building. ADME studies are unique and clients often have questions about them. The first question is how much radio-labeled material should we send? This will be determined by a couple of different factors, including the dose that is going to be administered and the number of subjects. The dose will be determined from the human dosimetry report, which extrapolates the dose from the animal QWBA studies. Celerion can write the human dosimetry report if needed. Standard ADME studies enroll 6 to 8 subjects. A rule of thumb is the amount of radio-labeled material needed is somewhere between 2 to 5 millicuries, which will cover both the mock prep as well as the clinical trial dosing. The specific activity of the material should not exceed more than 120 microcuries per milligram, as it simply gets too difficult to weigh once it's hotter than that. Another common question is, can Celerion help with formulation? Yes, we can. We have a lot of experience with different types of formulations in this field. We do need some sort of starting point, some sort of formulation, and solubility information is very helpful. Finally, can Celerion use leftover API from animal studies? Yes, that is also possible as long as the appropriate certificate of analysis is available. Often, repurification is required for this type of drug. Thank you again for joining me today. I have listed our pharmacists at all 3 of our global sites, any of whom would be happy to help with any of your compounding needs. At this point, I would like to open it up for any questions that anyone may have.
Well, thank you very much for that insightful presentation. I would like to invite our audience to continue sending their questions or comments right now using the questions window for the Q&A portion of this webinar. Now, I've already received some questions, so we'll get ourselves started with those. Dr. Foster, the very first question we have, I'm wondering from you if you can answer is, can Celerion also do the development work?
Yeah, so as far as the development work, we can help with formulation, as I mentioned. So basically, we are not formulation scientists. However, we certainly can help with those processes. We do need a little bit of information to start with. You know, we need the solubility information. We need some sort of starting ground. And then we can help as far as, does it work in this kind of a vehicle? Does it work with this? That being said, it's pretty primitive and that kind of thing, but we definitely, especially with the ADME studies, do that a lot. We do have the homogenizers and the mixers and that kind of thing, so it is something that we do help with quite a bit.
Very good, excellent. Thank you so much. The next question we have was wondering if you can comment on if there's any USP guidelines for ophthalmic products, or do they just need to use one for IV?
Yeah, so ophthalmic products are listed in USP 797, so we would follow the same kind of formulation procedures that are listed in there. So it would require some sort of sterilization. Typically what we see at Celerion is use of a 0.2 micron filter for a final sterilized product. So in the same kind of beyond use dates, that kind of thing. We wouldn't probably have a preservative used here at Celerion. It would probably be a compounding, a sterilization, and an administration right away.
Very good, excellent, thank you. Next question we have here would like to know, do you only compound for phase 1 or any early phase 2 small population studies? Any considerations for the practical differences?
So we have run some phase 2 studies. The caveat is that what we do compound for needs to be administered here at Celerion in our building. We're not able to dispense off-site. We are not a distribution type pharmacy. So if the phase 2 study is running here at our site in Celerion, then yes, definitely we can compound here. And really there would be no difference between phase 1 and phase 2. It would follow all of the same kind of guidelines.
Wonderful, thank you. Next, we have someone asking, would you consider dispensing a specific weight of API and reconstituting with a vehicle for oral administration to be compounding?
Yeah, that's kind of an interesting question. So it would, in a typical pharmacy, it would be considered just reconstitution. In our world, it would be probably considered compounding because the API, chances are it's going to be considered hazardous because it hasn't been tested. So as far as fees and writing it up and the master formulation record, those kinds of things, we would consider it compounding. But in the true technical term, it would be considered reconstitution. But as far as all the documentation and all that kind of thing, it would be considered the same and taken care of the same way as all of our compounding studies.
Very good. Thank you for that answer. Next, we will have someone wondering how far in advance are study drugs prepared?
So typically, the sterile products are prepared on the day of dosing. So we don't really do things very far in advance at all. Sterility is our highest concern for the safety of our subjects. We dose mainly healthy, normal subjects here. So we come in early, we will prepare the dose, and then we'll administer it right away. That being said, once in a while, we'll do things ahead of time. If there's stability and sterility information available, we certainly can do that. as long as we're within the beyond use date of 797. For oral products, that is a little bit different. So depending on what the sponsor is able to provide us for beyond use date, sometimes we can have 24 hours, 48 hours, 72 hours, just depends on what is available from the sponsor. Depending on the complexity of the compounding, longer is better and how much we need. Say if you're trying to run a SAD study and a MAD, the multiple ascending dose and the single ascending dose at the same time, it gets to be a lot going on at the same time. And, with a limited staff and that kind of thing, you want to be efficient, both with your drug product as well as your staff, and making sure that you have the space and the time to be efficient. So at the end of the day, we will do whatever the sponsor needs us to do, but we always like to be efficient with our time too.
Excellent. Thank you for that very in-depth answer. Next, we have someone wondering about sponsors. Can they observe compounding in the compounding suites?
Absolutely, they can. We actually have a sponsor here right now in our compounding suite, in our sterile suite, watching a mock prep. So what we ask is, if you're going to watch in the sterile suite, you do need to be garbed, and we have a designated person who will help with the garbing and that kind of thing. The other option, if you don't want to be in the suite, we do have cameras available that you can see in the actual BSC from the outside. So you can see what's happening. There's no audio, so you can't hear, so that's not the best part of it. But if you want to be in there, certainly we have no problems. We enjoy having sponsors here to see how things go and able to observe and kind of help with the process as we go along.
Fantastic. Thank you very much. Next, we have someone asking, how often is your compounding suite certified? And what do you do if something fails?
Oh, right. So it is certified by an outside vendor every 6 months. If we do have an area that fails, we will investigate if we can figure out any reason why it may have failed. And really, the first thing we do is clean the area and then retest. So we will, if it's, say it's a surface sample that's failed, for instance, we will clean the area with both a sporicidal and an antimicrobial, and then we'll retest it and make sure it's passed at that point. We will also have the microbe identified to make sure it's not anything terrible, and then we will just continue compounding from there. We have had a few, positive surface samples once in a while, but nothing that is detrimental or terrible, that kind of thing. But compounding will be stopped in that particular area until the organism is identified and we can retest and make sure everything is good to go again.
Very good, thank you. Now, we have someone wondering about staff and specifically how often are staff recertified in USP?
So the staff is recertified every 6 months also. So they can, we do glove fingertip testing as well as media fill testing. So the designated person oversees that. And if there's any kind of failure in that, then it's the same kind of system. We will do retraining and then we will test them again. And they would not be allowed to compound during that time if there was a failure. But we do all that right on site, and we do have a global designated person, so she's able to help with training and that kind of thing. So that is something that is done quite often.
Sounds like some good measures in place. Next, we have someone wondering for an ADME study. Is there a required percentage for radiochemical purity, i.e., should it be more than 95% or 90%, etc.?
So first of all, we don't have an SOP on it, what it needs to be. What I have seen from sponsors and what is typical is 95% or above. Less than that, sponsors tend to worry about degradation and having impurities in there and not getting a good result from their study. So all C of A's that I have seen through my time here have been 95% and above.
Fantastic, thank you. Next question would like to know, what type of water do you use for non-sterile API solutions, and what is a good bud assigned?
Yeah, so the water that we typically use for these preparations is AquaPure. That's the brand name, and we use that because we can get a certificate of analysis with it, and then we can add that to our pharmacy documentation. As far as the BUD with those products, really it's sponsor dependent. And if the sponsor doesn't know, our SOP really allows up to 7 days for those kinds of things. That being said, very seldom do we go to 7 days. I would say 72 hours is very, very common. But most sponsors have a pretty good idea of what they want for the beyond use date.
Thank you very much for that answer. The next question we have asks, can you manipulate API into formulations and adjust dosage levels?
Yep, absolutely. So we do that a lot. So we weigh different types of API either into capsules. Um, so that's common, but probably not as common as the oral solutions and suspensions. So suspension solutions, that kind of thing, often what we'll do is make a large batch of it and then aliquot doses off by weight. The other way that that can be done is individual doses into individual vials, solution suspensions. That's not as consistent because everybody's going to have a little bit of a different weight. If we are filling capsules, then each capsule is filled individually. It's a very manual process, but each capsule is documented and you would know exactly which subject got which capsule and how much API they got. We can weigh small amounts. I would say it's comfortable to weigh down to about 5 milligrams. Under that, it takes a while. It can be done. It's just more tedious as far as, when you get down to about 1 milligram, it takes a while to do it, but it can be done. So anything above 5 is easy, I guess I would say, and below that is just more time consuming.
Very good. Excellent. Thank you very much for that very in-depth answer and for all of the answers. However, we have reached the end of the Q&A portion of the webinar today. Now, if you're going to attend to some of the questions, though, the team at Celerion will follow up with you, or if you have further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speaker for her time here today, Dr. Jennifer Foster. We hope that you all found the webinar informative. Have a great day, everyone, and thanks for coming.
Thank you.